DNase Seq is a genome-wide assay used to study chromatin accessibility and remodeling. Genomic DNA contains the genetic information for all eukaryotic organisms and is wrapped around 8 histone proteins to form a nucleosome. Each of these nucleosomes are connected by linker DNA. The structure and organization of chromatin can affect accessibility and therefore gene expression. Transcription factors are proteins that combine to accessible regions in the DNA and recruit RNA polymerase to transcribe genes to make RNA. Thus, for genes to be transcribed, the chromatin must be remodeled so that the transcriptional machinery can gain access. When chromatin is condensed, it is no longer accessible to transcription factors and RNA polymerase. This is a form of transcriptional repression because inaccessible genes are not being expressed. If condensed chromatin is treated with deoxyribonucleases such as DNase 1, it is largely resistant to this digestion, as DNase, much like transcription factors, cannot gain access to compacted nucleosomal DNA. However, transcriptionally active open chromatin is not protected from digestion, and the enzymes will cut the accessible DNA regions that lack nucleosomes. The digested fragments of DNA can then be converted to a DNA library and sequenced. This approach is known as DNase Seq. DNase Seq first requires the dissociation of tissue or cultured cells into single cells using enzymes, immediately followed by nuclei isolation using detergents to break open the cell membrane and permeabilize the nuclei, and finally centrifugation to collect the organelles. The chromatin in the nuclei will then be digested using DNase 1. DNase 1 is an endonuclease that hydrolyzes phosphodiester bonds of the DNA backbone. Once the double-stranded DNA has been cleaved, blunt ends or sticky ends will be produced, depending on whether manganese or magnesium ions are present in the digestion buffer. After a proteinase treatment to get rid of chromatin proteins, the digested DNA fragments are separated by their molecular size using an agarose gel. Larger molecules will travel a shorter distance than smaller products because it is more difficult for them to move through the gel pores. The DNA products can then be identified and purified by performing a DNA isolation. Following DNA extraction, the digested DNA can then be sequenced using the Illumina platform. To do so, the ends of DNA fragments are repaired and sequence-specific adapters are ligated to the DNA fragments, which allows attaching of the fragments to a sequencing flow cell and enables DNA amplification by bridge PCR to form clonal DNA clusters. The Illumina machine then reads the sequences of millions of DNA clusters in parallel as fluorescently labeled modified terminal nucleotides are added by DNA polymerase in the process of DNA sequencing by synthesis. The resulting FASTQ sequencing file contains millions of reads that are then bioinformatically trimmed to remove the adapter sequences and mapped to a reference genome sequence. When multiple trimmed reads map to the same chromosomal sequence forming a peak, that pinpoints the location of a DNase-accessible region in the genome. Such DNase-accessible sites usually correspond to regulatory cis elements recognized by transcription factors. A bioinformatic pipeline can then be employed to identify conserved transcription factor binding sites in these accessible cis-regulatory regions of the chromatin. The DNase Seq protocol typically takes about a week to get from a tissue sample to a ready-to-sequence Illumina library, run the sequencing machine, and do the basic bioinformatic analysis. DNase Seq is one of several methods available to molecular biologists to study chromatin accessibility, along with MNase Seq, FAIR-seq, and ATAC-seq. All of these methods provide genome-wide information about chromatin accessibility and are most informative when performed on 2 or more samples in parallel. Thanks for watching.